afternoon. My name is Martina Stevens, and today I will be talking about plasmid power. Now please note that is plasmid with an ID, not plasma with an A. Plasmid refers to DNA, while plasma refers to something that can be made with a grape in a microwave. That's a story for another time. During this presentation, I aim to cover background information, plasmid preparation and function, an experimental example, and practical applications. Beginning with background information, one important principle of bacteria is that they have the ability to be transformed. This was demonstrated by Fred Griffith in 1928 in an experiment using two types of bacteria, an S-type that caused death and an R-type that did not cause death. When the S-type was killed, it did not cause death, but when the killed S-type was mixed with the R-type, it did cause death, showing the R-type had changed. A plasmid can be defined as a circular piece of double-stranded DNA found in bacterial and yeast cells. It contains genes that help an organism survive but aren't necessary to daily survival. They remain separate in the cytoplasm, does not have to incorporate to the main DNA. One useful purpose of plasmids are to transfer these genes, which is a basis of recombinant DNA technology when done artificially. So it changes genes to meet needs. Here's an example in which the plasmid is inserted into the cell, and then a useful substance is synthesized. Three components are necessary to create a plasmid: the gene of interest, restriction enzymes, which cut the DNA like scissors into sticky and blunt ends, with the sticky being more stable, and DNA ligase, which seals it all together like glue to form the circle. Here's a better example of that, in which the restriction enzymes cut the DNA, sticky ends are exhibited, and then the DNA can be placed in the plasmid, sealed with ligase, and then the plasmid can insert the cell to synthesize a product like human growth hormone. A cell can take a plasmid up naturally or artificially, but regardless, the cell has to be competent or able to take it up. Natural methods include taking it from the environment or another cell, and artificial methods include heating it, shocking it, as well as using a gene gun or other mechanical methods. One issue is that plasmids require a lot of energy for their genes to be expressed. Therefore, an antibiotic resistance gene can be put in the plasmid, and then it can be grown on antibiotic-containing material. In this way, it can also be identified or marked from cells that did not take up the plasmid. In review, this paperclip circle represents a plasmid, the gold represents the gene of interest, and the white represents antibiotic resistance. It can then be inserted into the cell, and a useful product can be synthesized. One experiment can be that the plasmids can be placed into E. coli cells, and then they can be grown on different plates containing different material, and the results observed. Two genes are present, an A gene with ampicillin resistance, and a B gene which causes sugar to be broken down in the colonies to exhibit a blue color. 6 plates are used in 3 different types of medium, and we see there is bacteria that contain plasmids as well as bacteria that do not. We expect growth on plates 1 and 2, 4 and 6, but not on 3 and 5. So to initiate the experiment, we have the 2 tubes, one that will contain the plasmid and one that does not. Calcium chloride is added to both tubes, as well as E. coli or bacterial cells, and the plasmid is added to the plus plasmid tube. Tubes are put on ice and then heat shocked to cause the plasmid to be taken up. After that, urea broth is added to provide nutrients, and then the cells are put on the 6 different plates. They are incubated for 24 to 36 hours, and then the results can be observed and the growth can be seen. Here are plates 1 and 2. As you can see, there is growth on both plates. There's also growth on this negative plasmid. It's hard to see, but it covers the entire plate in a bacterial lawn. And we expect that because the only thing in it is urea broth to provide nutrients. There's no antibiotic. Plates 3 and 4, however, are a different story. There's no growth on the negative plasmid plate because there's antibiotic, ampicillin added. However, there is growth on the plus plasmid plate because it has the antibiotic-resistant gene in it and it can grow on that medium. Plates 5 and 6 exhibit a similar story. There is no growth in the negative plasmid plate. However, there is growth in the positive plasmid plate, and it's blue because X-gout has been added, which is a chemical that can be broken down, or sugar that can be broken down to exhibit the blue color. Plasmids can be used to produce human products such as insulin, and benefits of doing this are that it can be safer, more effective, and create a greater product availability than other methods may provide. Other useful purposes for plasmid include gene therapy, human organ and tissue transplantation, which can take an animal organ and make it more similar to a human organ to minimize rejection, cloning, and new uses are still being discovered. In conclusion, in this presentation, we looked at background information such as what a plasmid is, preparation and function, and experimental example, as well as practical applications. Thank you for your attention, and at this time I would be more than willing to answer any questions.